So, thank you very much. I'll have the challenging talk before lunch, but I'll try to maintain the attention levels as high as possible. So I'm Panos. I'm working at the Globe Institute of University of Copenhagen at the Computational Biodiscovery Group with Thomas. And today I'm going to talk to you about our analysis on Streptococcus pyogenes and the phage-based approaches that we want to use in order to combat this problem in soft tissue necrotizing infections. So I'll start with a brief introduction on how the problem looks. Unfortunately, we have seen a surge globally in these clinical manifestations of Streptococcus pyogenes, or simply called Strep A infections. And we are talking about the invasive forms. We are not only talking about the sore throats and pharyngitis that we are most of us familiar with, but we are also talking about some more severe cases. So unfortunately, when it goes really bad in soft skin infections, it can look like this. And here we are talking about intensive care units. We are talking about amputations, and eventually, sometimes, unfortunately, even death, because the infection goes down to here and it becomes more systemic. If we take a look at the virulence pathways of this particular pathogen, here we can see a rather comprehensive figure. We're not going to go through that, of course, but I would like your attention here in a couple of categories, the DNases and the superantigens. And I'm saying that because these two categories of virulence factors are mostly prophage encoded. Especially the superantigens are actually responsible for causing what's called a cytokine storm. We had some quite nice immunology talks, so I'm not going to expand a lot. I will just explain a little bit how this works. So the superantigen here is responsible for bringing together this major histocompatibility complex number 2, which is on the APC, the antigen presenting cells, together with the T cell receptors in a non-specific, in a non-antigen specific way. So that actually releases a ton of cytokines, and eventually sometimes it can cause systemic failure or multi-organ failure and potentially death. So the Streptococcus pyogenes genome is from a pangenomic perspective is quite open. So that seems that we have a lot of mobile genetic elements that are actually inside the genome of Streptococcus pyogenes, and we're talking about prophages and phage satellites. So with prophages, we are very much familiar. They are able to integrate and induce autonomously, but the phage satellites are actually dependent on another helper prophage for them to complete their cycle, because they are lacking, most of them, they lack structural genes. So by living inside the genome of the Streptococcus, they have certain features that they can offer to the bacterial cell. And that brings us to the second part, which is the bioinformatic analysis of Streptococcus pyogenes. We wanted to understand this complexity as much as possible before moving to the control strategies. So we have established two databases, one with RefSeq genomes, a relatively small one, about 2,000 genomes, and a bigger one of 48,000 genomes. The first one, of course, is more curated, more robust. And the second one is a little bit on the content level, a little bit less complete with metadata. But after looking at both these databases, we saw actually the picture of the open pan genome quite well. So you can see, especially on the second graph here, that the accessory and unique genes are rather high, which actually brings the core genome quite low. And that means actually that the more genomes we have of Strep A, the more complexity we can add to the entire pan genome. This is also reflected here in the accumulation curves. But we can see that they are actually have the trend to increase. The higher number of genomes is included, the higher complexity is introduced, and especially in the SRA database, in the bigger one, it's even more steep, this curve. So having said that, we have developed an MLST method, which is basically, it's mostly based on the PubMLST database. And we have categorized our genomes, our databases, into different STs, like sequence types. And we have been adding to that methodology several, about 20 virulence factors of Streptococcus pyogenes. So in this case, I'm going to just mention a brief overview of the STs. As you can see, in both databases, RefSec and SRA, the most prevalent STs are actually in the top positions, and that's actually quite nice because we wanted to aim to the scalability of our strategy. And then following more on the evolutionary history of these genomes, we picked these representatives of the STs, and we constructed some phylogenetic trees. So this one is for the RefSec, and as you can see, you have two different ST types as the most prevalent across the genomes. And it's interesting to notice that the first one, the most prevalent, is actually very close to the common ancestor of all the Strep8 genomes, which means actually that it is still there, present, but then it has also been quite a lot diversified in the rest of the branches. And as I mentioned before, the superantigens are mostly prophage encoded. So we have included 8 of them in our analysis and MLST methods, and we wanted to see where do these fall in the branches. And if you see, they were there since the beginning, but then they are quite mobile, and they're distributed across all the branches. The similar picture we observed, a little bit more complex, of course, because of the volume of the database, but we observed similar patterns here in the SRA database. We have a lot of prevalent STs, which are, again, are distributed across the phylogeny. And then also the phage originated genes, the superantigens, are also prevalent across the entire phylogeny here. So going a further one step, we would like to analyze if we can connect, let's say, the metadata and clinical manifestations to the presence of these prophage-oriented superantigens. What we can see here, and it's quite interesting, is that the absence of the superantigens, and that's noted by the 0 on the y-axis, leads to less, let's say, clinical manifestations as they were submitted in the metadata. So that means that the higher amount or the higher number of superantigens, the more pictures like invasive diseases or invasive infections, they are a little bit smaller, so you cannot see it, but the more clinical manifestations like that are observed across the databases. And if we take a look at the skin infections, or one of the categories, we can see that this pattern is also validated. So the higher superantigens, the more cases that we observe of these particular clinical pictures. We should not forget that geographic locations plays a role, because different people who submit the data are from specific cases or from specific projects. So it is still something to take into consideration when we do this analysis, but they pretty much fit the patterns that we observe in similar clinical manifestations. So having analyzed the entire data sets in terms of mobile genetic elements, we found about 5.7 thousand of them in the RefSec database, and of course, 163,000 in the SRA. And here, what I want to show is that we have, of course, the phages, the prophages, we have the satellites, but we also have a category that I called phage satellites. And why is that? As I mentioned before, the satellites lack structural genes, and they depend on prophages. And here you can see the distribution of the top clusters, of the top groups. And on the y-axis, you see the closest relationship to the NCBI available data, genome accession numbers, actually. So the fact is that the satellites that mainly lack structural genes, it was recently published that there is a category called CFPK, or capsid-forming PKs, so capsid-forming satellites, that are able actually to have some structural genes in their genome. And the authors here, the Souza et al., published also some families that they found, several of them. So the CFPKs are present in Streptococci, but not as high as in other families, but still they were there. So in this publication, we took their sequence of type B and C, and we want to see if they're present in our databases. And in fact, they are quite difficult to discriminate from the phages because they still carry some structural genes, not all of them, of course, because they lack some tail proteins, so they are still not forming viable virions. But if we take a look at the matches that we found and the patterns that we found, let's say the type B, for example, we can see that this CFP is actually consists of the structural module that you can see over here, and then this other module of mostly hypothetical proteins. And as you can see, these both modules are present in several phage genomes. And what's in between are actually other genes, such as, for instance, virulence factors, like the super antigens that I mentioned before. So we can see that is actually a high modularity of these elements, and the similar pattern, similar observation was made for the type C CFPs. So yeah, so that being said, the prevalence of the superantigens was quite high. We did some clustering of all the phages and satellites across the databases, and we saw a very high modularity. Some of the groups were very consistent in terms of size, as you see this one, but then you have other groups like this one, where there were different fractions, different sizes of phages in the same particular group. And in order to highlight, it was shown actually how important the superantigens are, because in the top clusters, SPEA, which is one of the most prevalent, was actually quite evident in several of the top clusters of the mobile genetic elements. So they're quite prevalent and highly abundant, and it is indeed relates to the increased virulence of Strep A. So having a better idea of the structure of the databases and the bacteria and phages, we wanted to see what we can do in order to combat the strep pyogenes. We had a hint, and we already knew that it is quite difficult to isolate lytic phages against this species, also some others, but this is one of them. But we did our best. In the literature, we have seen that most of the phages used are coming from quite long time ago, but we wanted to build our own database, establish a biobank, and also sample in order to find phages. So we have been collaborating with hospitals, and we have been getting clinical strains from necrotizing soft tissue infections to build our own biobank, and we have already several of them. And we did a lot of samples from wastewater treatment plants and also wastewaters from different urban setups from Denmark, UK, and Japan in order to try to find phages. It was quite tricky, but eventually we found something. We then wanted, of course, to see what's there. We sequenced them, and I will talk about first the Danish phage. So this is actually a quite rare phage, although it showed some similarity to some known phages. It was not very commonly available in the NCBI database, and also it was not common at all inside the bacteria of Streptococcus pyogenes in the database. So we found an integrase in the genome, which means it's a temperate phage, and we also did the analysis of the genome itself. So this is the annotated genome, is an ISCFO. And then moving to the next candidate from Japan, we had also managed to sequence the genome, and we saw that it is a little bit less different than the available genomes, but it's a little bit more prevalent in the sense that it has more matches across the databases. But still, not so common inside Streptococcus pyogenes. And the family of this virus, Brucell is a little bit more common to the Streptococcus thermophilus, for instance. So again, trying to cluster it with similar phages, we identified that it is probably a temperate phage, and then also not a good phage for therapy. Here you see again the genomic analysis is also another CIFO, and the whole genome there is sequenced. So we kind of expected that, so we had a plan B, and the plan B has to do with endolysins. So this is like an alternative approach we want to use, and using the data and the knowledge we have based on the bioinformatic analysis and dataset, we could actually focus on the prophage content of Strep A, and then identify potential candidate of endolysins in silico, and move forward to the experimental setup and see what we can get. So this is a very nice picture from the Nelson lab of an endolysine killing a bacteria. Very briefly for the endolysins, that they are consisted of some protein domains, mainly enzymatic activity domains and cell binding domains. So I'm not going to refer a lot to that. They are part of the phage lytic cycle. So in the end, they are able to disrupt the cell wall. And in Streptococcus pyogenes can be quite unique. In our case, we focused on the PLY-PY because it's a one gene encoded endolysin. And it's also, I would say, simple kind of endolysin because it has only cell binding domain and enzymatic activity domain. And we wanted to identify out of our candidates which are their domains, and then how to utilize them in the experiment. So we did an analysis of the domains of our candidates, and we also clustered them in the sense of diversity inside each group. So we wanted to aim for the least diversity so that we can have a stable length of the same protein. And we ended up using some candidates from this particular family, which is actually a graphic representation here. You can see that they have like an enzymatic activity domain there and a cell binding domain over there. Is about 250 amino acids. So we selected the 5 candidates from this family, and this is also still work in progress. So these are more preliminary results, but we were able to. So what we did is that we made these 5 endolysins, and then we wanted to put them in plasmids, then the plasmids in proper vectors, and then we wanted to basically express them and induce their transcription, and then get the proteins themselves. We went through some purification cycles and did some work on that. And then we wanted to test their activity against our bacterial candidates, like the strep pyogenes clinical strains and collection strains. The picture was like that, more or less. You can see that these spots are actually showing the inhibition of the bacterial growth. So 2 of them seem to have worked. If we put them in a heat map, we will see that number 3 was quite efficient against all the strains. And these were actually clinical strains and also collection strains from different places. Number 2 was also following with relatively good performance. And then the rest, like 1 very slightly, but then 4 and 5 didn't really work. So in fact, out of this experiment, we concluded that there is some certain potential with this endolysine approach, and we need to further dig into that pathway for the STREP-A. So to summarize, the main points that I would like to raise here are that Strep pyogenes has an open pangenome with a very large diversity of mobile genetic elements. We have seen that the superantigens are highly prevalent in all the different types of Strep-A, and they're present very early in their evolution. We also saw that phages and satellites are quite highly modular, with several regions that are conserved and recombined around the species. And although Strep-A is a difficult target for lytic phages, we were able to still isolate some, which were temperate, but also come up with the alternative strategy of endolysins, which has shown some relatively promising results for the future. And last but not least, our in silico pipeline for the genetic element discovery, but also analysis, is something that can be scaled from a smaller database to a larger database, and of course, across different bacterial species. And with that, I would like to thank you for your attention, and also especially thanks to Rodrigo and Hannah, which we have been working quite closely on this project. They couldn't make it here. And also all the very nice colleagues and partners in this project, and the foundation bodies that subsidize the research. Thanks.